Good afternoon, everybody. We are very pleased to start with our fifth webinar of the immunomodel construction. And I'm going to give the floor to Professor McCormack, who will introduce the panelists. Please, Professor McCormack, go ahead and enjoy the webinar.
Thank you very much, Rosalinda. Good afternoon, everybody, and welcome to this webinar number 5, which is entitled Preclinical Modeling for Immuno-oncology. And it's an industrial perspective from toxicology all the way through to IND. I'm very, very happy to be able to introduce an industry faculty today. And I would like to thank the speakers very much for agreeing to come and speak with us today, sharing their experiences and their experience with industrial development of immunotherapeutics. We have Michael Boice this morning first, who comes from Certis Oncology, which is a preclinical CRO based in the Bay Area. We have Dr. Hilary Millar Quinn from Century Therapeutics on the east coast of the USA. And representing Europe, we have Katarzyna Wnuk-Lipinska from Novartis Biomedical Research in Basel in Switzerland. So the format for today, I would like to just introduce the speakers, give you a little bit of rundown on their background. The speakers will give their presentations for approximately 20 minutes or so. And I would ask if you have any questions for the speakers to please ask them in the chat. We'll try to get through one or two at the end of each speaker's presentation. And then with the time that we have left at the end, we would like to have a roundtable discussion with the panelists and open up the questions then from the chat. So if you have questions, please register in the chat. I'll try to get to as many of them as possible before we finish our webinar today. So before, without further ado, I would like to introduce Michael Boice, who is the Senior Director in Scientific Engagement at Certis Oncology Solutions, and he works directly with clients to enhance their discovery efforts through translational science discussions, optimizing the customer experience at Certis. He's based in the Bay Area and holds a PhD in pharmacology from the Weill Cornell Graduate School of Medical Sciences at Memorial Sloan Kettering Cancer Center, and he has over 20 years of experience in translational oncology, drug development, from bench science to outside sales. His areas of scientific expertise include the development of novel oncology therapeutics, functional genomics, and rare cancers, and he has an exceptional talent for strategic marketing and sales execution while managing cross-functional teams. So I would like to give the floor now to Michael, and again, remind everybody who's online to please send questions in the chat. Thank you very much. The floor is yours, Michael.
Thank you, Emmett, for that introduction. Thank you for everyone taking the time to be here today. Happy to be on the panel and present. And as Emmett mentioned, I am from Certis Oncology, which is a preclinical CRO provider that is based in North America, in California specifically. We service large pharmaceutical companies all the way to small biotech companies, and we are 100% focused in oncology development and oncology therapeutics. So we work with a number of different companies that have a number of different modalities, ranging from small molecules all the way to obviously through the topic of today's webinar, which is immuno-oncology. So we have worked with a lot of companies that develop immuno-oncology agents, and with that we have tried to develop different models and therapeutics that might be most beneficial for the different types of therapies that they're looking to develop in the immuno-oncology space. And so today, for the next 20 minutes or so, I'm going to talk a little bit about how we are working on our pursuit of precision using preclinical development of immuno-oncology therapeutics in humanized mice. So I guess just to lay out the landscape of why we are interested in doing this and why Certis is part of Certis' missions too is why are we doing this and what is going on. So there has been a lot of excitement in immuno-oncology agents over the past decade or so. Since 2011 with the first approval back in melanoma, there have been since then subsequent about 11 different approvals for immune checkpoint inhibitors that are now first-line therapies in a number of different oncology indications. Most of these are in combination with other different agents like chemotherapies and things like that, but they do range a wide variety of cancers too, starting with melanoma, moving on to non-small cell lung cancer and small cell lung cancer, and breast cancer now as well too, head and neck, and gastric cancer. So there has been a lot of excitement around checkpoint inhibitors and immunotherapy in general over the last 11 years or so. The problem with that is, though, we've seen that the overall response rate is somewhat limited in terms of patients that respond to this. We're talking about a response rate of 15 to 20%. Meaning that it works well in some of the patients, and we've seen some durable and long-lasting responses in a fraction of patients. But overall, we're looking at about 80 to 85% of the patients where IO drugs are not working well as they are in others. And the question really is, why is that? I know this is a very complicated question, and it is maybe not just one specific answer. It has probably to do with a lot of different things. But one thing that we're focused on, particularly at Certis, is because we feel like that preclinical models don't accurately recapitulate the human disease. And the fact that we're working with models that are inaccurate to predict response in humans is part of the reason why we're having such low success rate in the clinic with some of these immune checkpoint inhibitors as well too. So we are working at Certis to really kind of develop models that recapitulate human disease in a better setting that can be used not only for small molecule therapies and other different types of agents, but more importantly for immuno-oncology agents as well too. And so kind of just going over a little bit of the conventional models that are out there and what people are using to date. So obviously one of the ones that stands out as the most used preclinical model really are the syngeneic models, the carcinogen-induced models that really are popular amongst immunotherapy because they've been used for such a long time, and they have other benefits as well too, namely the fact that they are cheap when it comes to developing them. They are also cheap to use because they don't require immunodeficient mice. They just use your standard Black 6 or BALB/c mouse, and so they're much more efficient to use from a cost perspective. They're easy to use as well too, because they grow as a cell line. They are very easy to grow, to get implanted, and to run experiments, even large experiments as well too. They're also very easy to work with because they're easy to mimic as well too, right? So you can tailor these specific syngeneic models or cancer models to express different targets, and use that for different therapies and different modalities that you might have. That you're looking for as well too. Some of the things that make syngeneic models not so good as therapy or as models to use when looking at IO therapies is that you are looking at a lack of genetic complexity with these because they are cell lines that have been derived from carcinogen-induced middles. They lack the genetic heterogeneity that is seen in human tumor models. They also lack the ability to have the complex disease that is formed from human diseases as well too. So, and another thing that is we'll touch on later as well too is that most of the time these models are implanted subcutaneously, and so you're getting a readout that looks at subcutaneous tumor readout as compared to where you'd actually see the tumor growth. So, some of the downsides of those models are kind of listed there. Another model that has been used recently and has maybe gained a little bit more enthusiasm in the last few years is these genetically engineered mouse models, or GEMs. And these are a little bit different from syngeneic models in that they are genetically manipulated to induce cancers. And so they do have a little bit more benefits in terms of genomic and tumor heterogeneity that you'd see in a human tumor as well too. Some of the other downsides of GEM models is though that one of the big downsides of GEM models is that they are slow to develop and you can't really predict latency and or penetrance as well too. So fielding a study that would be used, for example, to do a pharmacology study in vivo is extremely challenging to use in these models. And then lastly, going back to what we talked about a little bit with syngeneic models, is most of these tumor models are implanted subcutaneously. And this is very challenging in that the fact that it doesn't really recapitulate human tumors very well, both phenotypically and genetically. And you're also putting it in a place where the tumor is not taken from or is not surrounded by the complex dynamic microenvironment that takes place when you're looking at micro or environmental oncology agents or immuno-oncology agents as well, too. So these are just some of the conventional models that are most oftenly used and the implantation sites that are most oftenly used in the conventional models. And what we're at Certis is trying to promote, and what we are really thinking about is how can we develop advanced cancer models that really might be better for answering the immuno-oncology question in terms of this dynamic interplay that takes place between not only the tumor cells, but also the tumor microenvironment and the stromal interactions that are required for a lot of the mechanism of actions that go along with immuno-oncology agents. And so one thing that we're really trying to promote at Certis is using orthotopic implantation of models, and for a number of different reasons. One is that it really does preserve the histological architecture and stroma construction, right, of the tumor and the microenvironment that it's placed in. It also has been shown to be a better predictor of not only response and resistance, but it also allows the tumor to grow more in the location where it was actually taken from and be in line with what you'd actually see in a clinical candidate as well, too. And then how can we do this better with advanced models as well, too? So we are really kind of trying to combine both orthotopic as well as humanized models because we feel like that those have the best chance of really showing the impact that you'd see in clinical samples as well, too. So when we talk about humanized models, we're talking about two different types of models that we're working with here, both PBMC engrafted or CD34 engrafted. So both PBMC engrafted and CD34 engrafted humanized models with PDX models implanted into them, we really feel like has the opportunity to define better outcomes in patients and more representative of the human tumor and the human disease that we see when we're specifically talking about oncology. And so we are working on using humanized models and PDX-derived models to really capture the genetic heterogeneity and the patient heterogeneity in which you'd see when kind of developing an immuno-oncology agent and moving into the clinic. And so, yeah, it's kind of getting into this question. You know, I've talked a little about in kind of hand-waving motions in terms of, you know, how does implantation site really affect recruitment and efficacy when you're talking about IO models? And does the implantation site matter? Well, that was one of the questions that we really tried to understand initially when we began using our preclinical models for our pharmaceutical and biopharmaceutical partners here at Certis. And so one of the first kind of experiments that we did here is really kind of understanding, does implantation site matter? When you're talking about efficacy and recruitment as well, too. And so we did kind of the obvious simple experiment that we have where we compared subcutaneous PDX implantation with orthotopic PDX implantation. And so we took a PDX model from our bank that was a colorectal adenocarcinoma patient that was taken from a 75-year-old male. It was originally derived as a CRC, but had liver metastasis as well, too. And you can see from these different mutations that it had a pretty wide variety of mutations that were, you know, hotspot mutations that really drive this cancer in KRAS, P53, PIK3, APC, and JAK1 mutations. And we did really kind of the obvious experiment here. We took this tumor, we implanted it both subcutaneously and orthotopically in eight mice. And then we treated it with a combination regimen of either FOLFIRI or FOLFOX in our mouse PDXs to see what the result would be. And interestingly enough, and probably not striking enough based off of this talk and what I've talked about so far, is that when we looked at these two tumors implantation sites, both subcutaneously and orthotopically, we did see a difference in terms of response to both the chemotherapy regimens. So, you know, not even thinking about an immuno-oncology agent, but just thinking about a chemotherapy agent. our regimen, we did see that there was a significant difference in terms of location or implantation site to how these tumors respond. So over here on the left, you're really seeing the subcutaneous tumor growth curve, whereas we had the control curve, and then we did see about a 30 to 40% reduction in terms of TGI for both FOLFIRI and FOLFOX. Now, when you compare that to the orthotopic implantation site, we saw something that was a little bit stark and a little bit different in terms of response. Whereas we saw a much greater response in terms of TGI with FOLFOX, but also we saw an even better response with FOLFIRI in the clinic as well, or in this PDX models as well too. So, not even considering the orthotopic or immuno-oncology implications of this experiment, we did see a heavy difference in terms of response to chemotherapy agents. And what's interesting about this is that we do have kind of a clinical arm to our business as well, too, and that we were able to take this patient's tumor, grow it up and do this experiment, and then show the oncologist and the patient how well it responded to FOLFIRI in these mouse PDX models. And then we went back to the oncologist and showed them that the tumor itself had a really good response to FOLFIRI, and the patient actually did go on to receive FOLFIRI in the clinic and had a similar response to what we see here in the PDX models. So obviously this led us to the question of, we see this information, see the difference in terms of implantation site in regular studies, just using chemotherapy agents. What would this look like if we were to do kind of a similar study using an IO study, implantation in immuno-oncology agents as well too. So again, we took a similar type of setup where we used one of our unique PDX models, this time from a primary gastric adenocarcinoma from a 76-year-old male. Again, you can see it did have some prior treatment, mostly chemotherapy agents this time. And we did kind of the similar experiment that we did before, except this time we did something a little bit different to make it more humanized as well, too. So we did put the tumor in 2 different locations again, too. We implanted some subcutaneously, some orthotopically in the stomach. And then here's where we did the humanization as well, too. So we've done some prior characterization with humanization showing, the level of humanization that we would require and optimizing the number of cells that we would require from a donor PBMC. And then we really wanted to look at a couple different things to see how they compare, looking at survival of the mice, also looking at the percent of humanization by CD45 chimerism, and then also taking a look at peripheral leukocytes, as well as tumor-infiltrating lymphocytes to see if we could see a difference in terms of implantation site in this particular study as well. Oops, sorry, wrong way. Sorry about that. Okay. So what did that look like overall? So looking at our first kind of outreach, what we were looking for here is, and sorry, my graph is not labeled there, so let me just pull up both these graphs here. And so what we could see here is that we're implanting both sub-Q using a vehicle as well as PBMC listed by the black and red, and then using orthotopic, both vehicle and PBMC injection as well too. So what we can see here from the survival curve on the left is that really there wasn't much of a difference in terms of survival between the 2 different implantation sites. Both subcutaneous vehicle and PBMC injection, as well as orthotopic vehicle and PBMC injection, really kind of gave a median time of survival of about 40 days. And that really is in line with what we've seen before with our experiments in terms of optimization when we're doing these humanization experiments. And then how did it look from a humanization perspective as well, too? So as you can see by the black and blue curves here on the bottom, the vehicle showed no humanization, whereas we're looking at the mouse humanization, or the humanization in the mouse blood. What we see is that both the subcutaneous and orthotopic humanization really kind of reached a level of about 40% or so after 40 days. Really kind of the optimal level of what we'd like to get to in terms of humanization. So there was very little difference in terms of survival and humanization when looking at either subcutaneous or orthotopic implantations. And now looking at the last 2 things that we were interested in looking at is also kind of understanding the circulation levels and the TIL infiltration levels of humanization as well too. So initially looking at in the blood, looking at overall CD45 percentage, and then also looking at individual T cell concentrations as well too, looking at different populations from CD3, CD4, and CD8. What we can see from the blood is that there was really very little difference across these populations from an overall CD45, CD3, CD4, and CD8 percentage in the blood. Comparing that to what we saw in the tumor, where we did see a significant difference, in fact. So in the tumor, the percentage of CD45 cells that were able to infiltrate in the tumor was much, much less in the orthotopic implantation site compared to the subcutaneous site. And this was different not only in CD45, but also a difference in CD3, CD4, and CD8 cells. And so what we're seeing here is that when we implant tumors orthotopically, the level of infiltration that we see orthotopically of these T cells is really different and a lot lower when comparing subcutaneous and orthotopically. So this just gives rise to the fact that the implantation site does in fact make a difference. And so some of the efficacy results that you might be seeing when looking at subcutaneous might not also be available looking at orthotopically, just because of the amount of cells that are actually able to infiltrate the tumor in the orthotopic site compared to the subcutaneous site. And so obviously, kind of the next question that we had with this data is, how would that affect treatment with different IO agents? And so we set up a very similar type of experiment there, where we did subcutaneous and orthotopic implantation of the two different models, and then again, humanization. But this time we did treatment with anti-PD-1. And then this time we were looking at not so much survival and humanization, because we had a good handle on that, but we wanted to look at really see how this would impact, treatment would impact tumor growth, TIL expression, and then also looking at TIL recruitment and activation based off of the previous experiment that we did that showed it had a significant difference in terms of infiltration. And so we did, so we implanted the tumor both subcutaneously and orthotopically. And as you can see here from these MRI images on the left-hand side of the screen here, we can see a nice tumor growth, both subcutaneous, and then also looking at orthotopic tumor as well, IP. And again, when we looked and saw this, looking at the tumor growth specifically, we didn't see very much of a difference in terms of growth characteristics of the tumor compared from orthotopically to subcutaneously. The tumors grew about the same through 30 days, getting to about 1,000 millimeters cubed. And again, here we did see differences in terms of T cells per milligram per tumor. This time, surprisingly, we did see a little bit more T cells orthotopically compared to subcutaneously. But one thing that was actually different here is that we did see the activation of T cells was different compared to orthotopic, compared to subcutaneous. And so then again, we wanted to check and see how well these tumors would respond to IO treatment. And so what we can see looking at the tumor growth inhibition kind of curves here on the left is that subcutaneous tumors did have a little bit of growth inhibition, about a 30% TGI compared to an orthotopic tumor where we saw about 3% TGI, almost no impact in terms of tumor growth. And then again, looking at the intratumoral cells that we were interested in evaluating, we could see a significant difference here. Again, not only in the number of PD-1 expressing CD4 positive cells, but then also in the number of CD3 cells that were induced after PD-1 compared to the controls. And then again, also implantation site made a difference in terms of activation using interferon gamma and granzyme B in terms of CD8, CD8 as well too. So where we had a significant difference in terms of activation of these cells in the subcutaneous setting, whereas we didn't see much difference or significant increase in these CD8 cells in the orthotopic setting as well too. So, I mean, overall, these two experiments really kind of laid out the foundation for us to believe that, implantation site really does make a difference when we're evaluating immuno-oncology agents. and why we're seeing a kind of a diminished response orthotopically. That could be of clinical significance in terms of evaluating immune checkpoint inhibitors and other immunotherapies as well, too. So, and we've also done similar experiments like this with CD34 engrafted humanization models as well, too. So, looking at a metastatic adenocarcinoma line that we've done that has extremely been previously treated as well, too, with not only IO agents also as well, too. And we can implant these different animals, different tumor models into different locations in the brain, lung, and the bone, and monitor these. I'm sorry, my graph isn't showing up for some reason, the labels as well too, but this red line is treated with PD-1, combination with PD-1 and CTLA-4. So we do have the ability of looking at these also in CD34 humanization models, not only just the PBMC ones, but we can also do the CD34 engrafted humanization models. And so really this kind of brings us to our flow when we're thinking about immuno-oncology translational workflow that we're able to work with. And because we have access to fresh patient tissue samples, we can actually do a couple different things here when we're working with them. And this is kind of the gold standard. What we would really hope to work with is taking both the biopsy that we're getting from patients, culture, and expand those TILs and use these TILs, these autologous TILs, for both co-culture treatment, looking at this, as well as in vivo characterization of an autologous tumor with infiltrated TILs that we get. And the other avenues that we can do is kind of what I've talked about so far as well, too, is looking at orthotopic implantation with PBMC humanization in our in vivo PDX model setting, looking at in vivo IO therapeutic efficacy. We can also, because we have these tumors, we can also look at genetic analysis, preserve these tumors, and also recall these tumors based off of genetic defects and genetic manipulations that might be of interest to people. And all this information that we have is housed in our cancer model database, where we can search for specific patient characteristics to make sure that we're choosing the right PDX model to look at humanization experiments. And so with that, I think my 20 minutes is about up. I will just leave this on the screen for a second, that if you have any questions or would like to get in touch, here is my contact information. And yeah, if you have any more information about learning about Certis as well too, here are some websites that would be of interest to you. And I'll turn it back over to you, Emmett. Thanks so much for paying attention.
Thanks very much, Michael, for that very nice overview and the differences between the subcutaneous and orthotopic models in particular. So we seem to be having a little bit of a problem with the question function. So I ask if anybody has questions to ask. Maybe you press the raise hand function and I will try to get you online. But I have some questions for myself. You mentioned in the humanization part of your model, where you establish the tumors either subcutaneously or orthotopic first before coming in with 5 million PBMCs. So I know from my own personal experience, that's quite different from what we would normally do, which is to humanize first and then put the tumors in. So I just wondered how soon after the tumor growth do you put in your PBMCs, or do you have to wait for a particular tumor volume, or what's the practicalities around that?
Yeah, the practicalities around that are actually, it's a very kind of tricky and nuanced procedure, just because we are when putting in PBMCs, the one disadvantage of that experiment is that you would have kind of graft versus host disease issues as well too. And so we kind of have to balance out how quickly the tumor grows versus how soon we implant the PBMCs. And so it's all understanding a little bit about how quickly the model grows from a tumor growth perspective, how quickly we expect it to grow, and then also trying to implant the PBMCs kind of before that tumor growth really takes off, so that we're not missing the treatment window that we'd like to see as well too. So it's a combination of, maximizing the PBMC donor information that we have and implantation, and then also allowing for particular time for tumor growth inhibition, and then also making sure we have that that therapeutic window as well too. So it is a little bit of a tricky interplay, and it really depends on the model that we're talking about and how quickly it grows.
Thank you very much, Michael. So I have another question here from Professor Dr. V.K. Ulegantanan. In your humanized PDX model, when measuring T cell infiltration and activation on tumor site, are you looking at specifically gating on human T cells, mouse T cells, or both?
Yeah, so that's a good question. So we are looking at specifically human T cells. So we do gate on human CD45 cells specifically initially to start, and then we go to previous subpopulations after that. But we are looking at humanization, human CD45 cells and gating out the mouse CD45 cells.
Okay, great.
And I should also say that these models are also in pretty immune deficient mice. So the level of mouse T cells and other mouse immune cells that are there are pretty limited depending on the background that we're using. So we shouldn't really see much mouse interaction. So that's why we're hoping to get mostly human interaction as well too. So
great. Thanks very much. Sam Chuang, do you want to turn on your microphone and ask your question?
Yes, can you hear me?
Yep.
Great. Thank you so much for a really great presentation from Charles River. In your first case scenario where you used the patient biopsy and then was able to actually relay the results to the clinician, that's pretty exciting to be able to direct the treatment. But how much time did you need in order from that biopsy to the time of designing the treatment that the patient had to wait?
Yeah, that's a good question. And again, it always depends on the tumor growth kinetics of the model as well, too, right? So it is a process that does take a little bit longer than we'd like to for some of these patients as well, too, because we're kind of up against fighting up against a clock to some extent. So for that particular patient, we were able to get results back relatively within a meaningful time frame, but we don't always have that time frame, which is why we're working on ways to make it faster. And so we have a different thing that I didn't really talk about today, but where we're using predictive AI to really kind of predict how therapies would respond to patients' tumors. And so we're using that now a little bit more as a proof of concept and a quicker mechanism for where we can get information back sooner to the clinicians and the patients to really make sure that we have actionable decisions faster than it takes to design and develop these PDX therapeutics. Because as you mentioned before, that is a long process that takes anywhere of a couple different months, and we want to make that turnaround time less. So yeah, we're working on kind of a predictive AI model to increase that time we're talking about there.
That's great. And sorry, I have one more follow-up question. Really fascinating with being in the in vivo pharmacology space. You showed a lot of great information where location of implantation was actually a critical factor. But in the drug development space where we're more screening, proof of concept of efficacy, we generally take a more standard approach. How would you translate the differential findings that you found and translate that to more formal drug development screening for identification of drugs or combination of drugs that could be commercially viable and more efficacious for patients?
Yeah, I think that's a good question. And always, what we're dealing with here is budgets and timelines and things like that. So the experiments that we kind of talked about here, we understand are expensive. And we also aren't under the illusion that syngeneic models and gem models do have their place because they are cheap and they are easier to run, right? And so I think you really have to think about when you're designing these experiments and which models you're using is to understand that these are all models and they can answer specific questions, right? So, when you're looking at, less inexpensive, more high throughput models, those are probably better when you're talking about early stage candidates, mechanism of action studies, things like that. But when you get into, really kind of the translational sciences of it, the translational part of it before you move into the clinical studies, that's where we really kind of think about, can you use some of these more advanced models, some of these more challenging models to see how well you actually would do, and maybe use that for translational information into the clinic.
Thank you very much, Michael. Unfortunately, I think we have to move on to our next speaker now. But Anna and Nuno, I've recorded your questions. If Martine or anybody else has questions towards Michael, please write them in the Q&A. or the chat, and I'll try to get to it at the end of the webinar. Thank you very much, Michael. Very much appreciate you getting up at the crack of dawn to join us today. And now I'd like to move on to our next speaker on the program here, which is Dr. Katarzyna Wnuk-Lipinska from Novartis Biomedical Research in Basel. Kasia has over 13 years of experience in oncology, drug discovery and development, and she has led pharmacology for oncology drug discovery projects spanning from target validation up to DC enabling and combination studies supporting clinical development. Kasia holds a PhD in biomedicine from the University of Bergen in Norway, and she currently works at Novartis Biomedical Research, located in Basel, Switzerland. She has previous experience from both biotech and pharma industry, where she has had the opportunity to contribute to scientific project progressions from early preclinical discovery up to first in human studies. And Kasia is now going to present her topic, which is development of immuno-oncology and therapeutics for preparation in IND. Kasia, welcome, and thank you very much for joining us today. And with that, you have the floor.
Thank you so much for the invitation. So as Emmett said, I'm Katarzyna Wnuk-Lipinska. I work currently at Novartis as a lab head in pharmacology, oncology, drug discovery. And what I'm going to present to you today is some concepts that are existing in the preclinical evaluation of small molecules in oncology drug discovery. So conceptually, there are different phases in drug discovery, whether it's preclinical or clinical, but also in the research, we actually can speak of the phases, so different phases, and they may be called differently between different organizations, but the concept is the same, and that with each stage of the preclinical research, there are toll gates keeping guard of the discovery effectiveness, because efficiency and also strategic fit. So, as you know, maybe the probability of successfully making it to approval from the non-clinical stage in oncology is around 4%, so it's very low, and it's quite risky business that we're in. And the mean drug development cost reaches about $85 million, data from 2018, but it's only the expected cost of actual program, which represents only research. But when we also adjust for the cost of failures, that cost increases to about $600 million. When we, on top of that, also capital cost, which also includes the infrastructure, that cost already exceeds over $1 billion. So this is definitely a difficult space, and therefore it's good to have some sort of guiding principles as to the evaluation criteria of each of the research programs focusing on drug discovery and development. That would allow us to make guided decisions across different phases of drug discovery. So there are components of the preclinical drug development that are considered essential to know, certainly prior regulatory filing, but also at different stages of the profiling of the molecule, in order to make an educated decision if the molecule actually has the right profile to be considered for, first of all, further optimization efforts, the second, which molecule to choose from if we have a set of molecules, and third, if it should be even considered for the full development later on. So these different concepts are not organized per phase, but within preclinical research, but rather per area of activity. And of course, logically, you would potentially start with physical properties, initially start with biochemical assays, and then you will go for in vitro assays, check in vitro potency. If that looks promising, maybe check the low dose PK, then expand to PD, etc. In the meantime, maybe check some components of the safety pharmacology, such as hERG, for example. But all depends on the target and knowledge that was built throughout the target validation efforts, throughout the assay development, but also based on the data that is available in the literature. What is important to know here is, or note, is that all these elements are essential and should be considered prior investing in the clinical readiness preparations and further early clinical development. So as we ought to be focusing here on the in vivo aspects and also on the small molecules in my case of the preclinical research, let's start about the importance of the PK. I'm assuming that most people in the audience actually are familiar with the PK itself, and everyone knows that PK is nothing more than what the body does to the drug in simple terms. There are several ADME components, again, to consider, such as absorption, distribution, metabolism, and elimination. And they are all equally important and have to be carefully evaluated while keeping in mind the nature of the target and the effect we want to achieve. So this is just a simple example of a PK profile of a compound administered orally, Barroso, per abbreviation TO. And my aim here is not to give a lecture on different elements of the PK profile, but rather on the importance and translatability of them. So, for example, Cmax, if you're expecting on target tox, you probably won't like to have high Cmax, right? Because this could potentially result in tox reaction. So now, another aspect is, for example, is the half-life sufficient to drive the efficacy? Another question would be, what is the elimination, as we also want to be sure that the drug can be washed out, and particularly in an event of unforeseen adverse event, particularly in the patient already. In oncology, frequently you want to have either high or constant exposure, and that leads me to my next point, which is that it's good to know the PK driver of the efficacy, or at least have a good suspicion what would that be. This can be typically assessed again in combination with the PD analysis, initially in vitro, followed by the in vivo assays. And this brings me to my next point, which is the pharmacodynamics. So PD represents what the drug does to the body, and as such, it helps to understand the drug-target interactions. We navigate through PD assessment with the use of biomarkers that reflect the pathway activation or inhibition. What is important, biomarkers can be indicative of both efficacy, but also of potential clinical benefit further down the line, but also unwanted toxicity. Now, PK and PD coexist together, and it's very important to understand their mutual relationship. And here are some examples of how this relationship could look like. So in the top left panel, we have a representation of the sort of most wanted sigmoidal profile, where the peak response and the peak concentration are correlated when they draw against the time. And just below, so here, this figure shows the concentration versus the response. So in the short, the response follows the drug concentration nearly in real time. And this relationship is very easy to navigate through in PK/PD screens in preclinical scenario, but also makes life much easier also in the clinic. Now, two other examples here in the top panel represent a scenario where the response is delayed. So the peak response and the peak concentrations are shifted, and here actually on the second graph, it's the shift is even more prominent. And when you look then at the relationship of the response and concentration, you will see that this dependency forms sort of a loop. So here in extreme situations, even when having no drug concentration in the system anymore, we can still see an effect. And this scenario can be particularly difficult to control in case of on-target toxicity. Now, PK/PD relationship is then further integrated with, of course, with efficacy and safety. And typically, biomarker modulation predicts efficacy. Now, observing PD modulation without apparent efficacy can mean that we have, for example, pathway activation, but the signal is potentially not strong enough to achieve therapeutic effect. Or potentially there are other components of the microenvironment or other pathways that needs to be taken care of in order to achieve benefit. By far, plasma concentration remains the best measure to correlate the drug level with outcomes. And this applies not only to efficacy, but also to toxicity, where the unwanted effect can be either on or off target. Now, to navigate through the efficacy and safety data and define the therapeutic window where the treatment would be effective but yet safe, we calculate the therapeutic index. Therapeutic index utilizes exposure-centric approach and typically focuses on the unbound values of the exposure. So this is simply an exposure at the safety endpoint over the exposure at the efficacy endpoint. And as we progress through the development program, the confidence in this metric increases because we generate more and more data. The molecule is being optimized for specificity. We understand better the PD efficacy relationship, and the modulation leading to our therapeutic effect improves. At the same time as the molecule is being optimized for specificity, we have less or none off-target effect. We generate data during the DRF study and later also GLP tox, and we know exactly what dose levels become toxic. Further, we can use allometric scaling principles and make prediction for the starting and efficacious dose for the first in human. So I have alluded to you some of the aspects of the drug discovery and development, and hopefully I was able to sort of see the sense of a great collaboration that is happening in the drug discovery. And it has to occur in order to make the program successful. It is certainly not a one-person job, neither can be done or navigated by one person. And you need to have experts coming from different functional groups. And each and every of these persons will have an integral and critical impact on the discovery program. And neither of these functions should be omitted. So collaboration is the key to success in drug discovery.
Ale wcześniej, tylko już ci powiem, ja będę u pani tej, a w ogóle jeszcze o tym też chwilę chciałem.
Someone should maybe mute. As a summary, there are several general things that have to be taken into account while embarking on the next steps within drug discovery and development. And one is identification of the right target and the right pathway. So how confident are we that modulating the specific signal is sufficient to treat cancer patients? Is the target druggable? Is it novel and brings an improvement compared to already available therapies? Another one is how strong does the signal has to be as a complete shutdown of the pathway required, or alternatively, what level of pathway induction could drive therapeutic effect? Also, in many situations, specific pathways either up or down regulated in many types of cancer, not only in one. So it is critical also to select the right patient population to start with the POC studies, so proof of concept studies. And we need to be sure that the therapeutic strategy we want to offer to the patients has the potential to actually improve currently available options. Also, we anticipate the effect to be as strong as possible, and in some indications, it can play a bigger role than in others. Now, further, do we have the resources or some sort of strategy to get them to commit to a full clinical development of the compound? This applies to the costs of the trial itself, but also time and human resources to have the trial designed thoroughly and appropriately. Are the selected endpoints right? Is the sample collection sufficient, but also not overly exhausting for patients? Will patients be willing to enroll to your trial, taking the trial design and potential benefit for them? Will physicians will be willing to enroll patients, then commit their time to your trial? And another one is, again, back to biomarkers. What kind of data will you collect? How confident you are that the PD efficacy relationship actually works? If using a surrogate PD readout, how confident we are that the readout actually translates into the clinical benefit? And at the end, I actually prepared a bit of a table with different values that are generated fairly early in the clinical development. And this is mock table, so this is not coming from a real program. So the resemblance is unintentional here, but let's try to look at it together. So for the compound 1, we have actually the biochemical IC50 reaching nearly 1 micromolar, so it's very high. It will be difficult to actually, based on this data and also based on cell line proliferation, here's actually GI50 used, would be difficult to reach appropriate levels in the clinic. So this molecular restarts do not look promising. Further, when we look at the selectivity for the isoform panel, we also see that there is actually 2 micromolar levels. So we actually have only 2-fold over the potency. Again, does not look promising. Exposure is almost not there. So the drug concentration is fairly low. And also safety pharmacology panel is, well, it's over 5 micromolar. Again, can be difficult to reach this kind of level in the clinic, but also on the other hand, it's actually only 5-fold over the potency. And when we look at hERG, actually it's for hERG IC50 is 1 micromolar, so it's pretty much exactly the same as cell line proliferation and thus IC50 in biochemical assay. So this just simply means that if we will be targeting, hitting our target, we would be also hitting hERG. So there is a very clear no-go for the compound 1. In case of compound 2, this one seems to be quite potent. We have a little bit over 200 nanomolar, so fairly okay. Cell line proliferation is about 70, so it's a little bit higher than for the biochemical assay. This will also differ between different cell lines and different models. Further selectivity for the isoform panel it looks very good. It's over 100 micromolar, so it does not raise any flags. AUC also looks very okay. Safety pharmacology, hERG, everything looks fine. We don't have TDIs, we don't have CYP inhibition. For the Ames genotoxicity assay, also we have negative results. The only small flag is that it's weakly positive for the phototoxicity assay, but this is also it's not a no-go. It just simply means that potentially in the clinical trial later on for the patients that will be using the drug, if successful actually at the end, would have to use SPF creams, potentially. Would have to be careful with the sun exposure. For the compound 3, most of the parameters look actually fairly promising. The only problem is that we have, again, here, the safety pharmacology in red. So we have a certain flag for hERG, and even though we actually have quite a big fold difference between hERG and the potency, still 1 micromolar is actually fairly low value for hERG, and it could actually bring some cardiac tox issues potentially later on in the clinic. So slight red flag. So with that, I'm going to end. I hope this was useful, and I'm happy to take questions.
Thank you very much, Kasia. Sorry for the slight delay there. We have a comment from Sam Chuang saying, great presentation. I'll just wait to see if there's any questions that come in on the Q&A, but I had some questions myself, Kasia. So it's a really nice presentation, particularly when it comes to small molecule inhibitors, when it comes to the immuno-oncology space. Would you run through the same types of protocols as you described there for a cell-based therapy, or would that follow a different trajectory?
Right, so I would say it follows slightly different. So there are some commonalities here, definitely. I also have to say that I'm not an expert in this field. I'm definitely a small molecule person, so I think we would have to also ask for advice of another expert. There are certain similarities. I would say maybe the selection of the models at the end is different. That definitely triggers some different approaches. Even PK as such is of course different as well. This is something that you definitely track differently. And then later on in the talks, you would also consider whether, for example, so for small molecules, typically you would consider rat and dog. For cell-based assay, you would maybe consider monkeys actually as the closer resemblance. I'm not sure whether Hilary is maybe not going to talk about this also a little bit more. I'm not sure. But yeah, I hope I helped, but I think that we should consult here also someone who is actually more of an expert in the cell-based therapies.
Thank you very much, Kasia. There's a question, Marcin, you have your hand up. Do you want to ask a question?
So yes, thank you. I had a question on, like, it's I would say slightly next to the whole discussion. It was also more about biologics. So how would you then assess in terms of this PK data, the PK of biologics? I mean, especially comparing the animal data and human data and the difference. And I mean mostly about antibodies or ADCs.
Right. So antibodies, you would simply run ELISA, right? And you would check the concentration differences. You can also do actually correlation of the exposures, compare exposures with the effect, right? Of course, you expect the PK will be completely different for antibodies, right? The profile is completely different than for small molecules, and the retention time is much longer, right? For the ADCs, it's a little bit more complex because you're actually dealing here with both small molecules and antibody, right? So it's again a little bit different approach. So again, I'm gonna have to send you to an expert that is actually dealing with the with antibody-based therapies.
Thank you very much, Kasia. Now we move on to our last speaker, and it's my great pleasure to be able to introduce Hilary Millar Quinn. She graduated with a Master of Science in Pharmacology from Thomas Jefferson University in Philadelphia. And Hilary has worked as Director of In Vivo Pharmacology at Century Therapeutics, a cell company focusing on the discovery and the development of products to treat oncology and autoimmune disease indications using engineered iPSC-derived effector cells. And Hilary has almost 25 years of industrial experience in preclinical in vivo modeling for the development of large and small molecules and cell therapies. Her experience includes several years running toxicology, in vivo studies at GLP environment, and 16 years at Johnson & Johnson supporting oncology-focused preclinical pharmacology efforts. She has a keen interest in drug development and post-IND translational activities and designs models and studies with an eye to further understanding drug mechanisms to advance the best candidates to IND. Hilary actively participates and presents in conferences such as AACR and STIC and is an author on numerous publications and several patents. So with that, I hand the floor to you, Hilary, and we're excited to hear what you've got to tell us.
Thank you so much, Emmett. Can you see my screen okay?
Yes, we can.
Okay, good. Actually, it kind of worked out really nicely because Michael's presentation and Kasa's presentation actually is going to be wrapped up a little bit in this presentation, and I can also answer questions about large molecule and cell-based therapies as well, because I think that's more of a specialty for me than the small molecule part. So yeah, I can try to address some presentation. So today I'm going to talk about the development of immuno-oncology therapies in preparation for IND. There we go. I'm going to bring up a quick disclaimer, and it's only because I know it's advertised I'm an employee of Century, but unfortunately, as of last week, I'm actually no longer an employee of Century Therapeutics. We had a round of layoffs, and I was part of that. So I did want to put that out there, but I am still active in this drug development community and looking for a position currently. And also the information in the slide deck does not necessarily represent the views of Century or any of my other former employers. So today I want to talk about drug candidate attributes in the IO space around target, mechanism of action, molecule, and cell type, and how all of those things go into developing a drug towards IND. I'll talk about assay selection. And again, this is a little bit more for the later preclinical stage when you're moving into IND enabling studies, whether to use in vitro or in vivo assays, and also the clinical translatability versus proof of concept. I'll touch on safety testing and then also some information for correspondence with the FDA in preparation for IND. So, I need to minimize that. Okay. So this is actually very similar to what Kasia was just showing, kind of a discovery to IND format. Up here, you have your molecule discovery, which includes target identification and validation of the target. You're going to generate your compounds and do some high throughput screening to see which ones bind the target the best. You'll have your hits, your top hits, and hit to lead, and then lead optimization. And you'll be going through preclinical testing, which assay development is usually, throughout here, but then you further refine for your preclinical in vitro testing, in vivo testing, and then choosing your development candidate. Typically, in my experience, most companies or most programs have chosen 2 candidates for development, and that is to have a lead and a backup. Usually throughout genomic testing, especially with cell therapies, or other testing, you find that maybe your lead shouldn't be your lead anymore. So you do have this backup candidate with maybe slightly different attributes. So that's a way to kind of mitigate that risk. And today, really what I'll be focusing on are IND-enabling type studies, which should be done with a GMP or GMP-like manufactured drug product. And the studies are going to include pharmacology, safety, and PK studies. And these should be done in a GLP or GLP-like setting. I will say that my experience has also said that at least for when you're starting off with a new class of drugs, the safety testing should be done GLP if possible. The FDA does like that. Once you are expanding on that class of drugs and doing developing more of those drugs downstream, you can do GLP-like. So this is kind of my triangle for IND. And it's really focused on the mechanism of action of your drug and the target. Associated with these 2 things are all of the rest of the work that you have to do: the safety and PK, the efficacy and PD, and then your proof of concept studies. So under your safety studies, you would be thinking about on-target off-tumor toxicity, cytokine release, acute toxicity, immunogenicity, and that could come, of course, from repeat dosing, and pharmacokinetics. Under the umbrella of efficacy and PD, you would have anti-tumor activity and proof of mechanism. And then for POC, this could be proof of concept for any additional features of your drug. I will focus a little bit more on this as it goes with cell therapy, because especially in the space that I am in or I was in, our cell products have multiple engineered features. So you do want to prove that those features are operating as designed. And then not only that, but that they are improving the efficacy if that's what is desired, and they are not exacerbating toxicity. So these are all interlinked. So some questions to guide you when you select your assays. Is my drug a small molecule, a large molecule, or a cell therapy? If it's a small molecule, is the formulation appropriate for murine studies? Because this is pretty much what you're going to have to use for your drug development, or mice or rats in some cases. So this goes with formulation. This goes with the actual construct of the chemical, and what's nice about the murine with small molecules in a murine setting is that you can use competent animals, which are have their full immune repertoire, and you can use syngeneic tumor models. So that way you can really see the interplay between the mechanism of your drug and the downstream efficacy of it in the tumor. So Tasha just went through all the background stuff that goes around using a small molecule, especially around the PK and PD, so I'm not going to get into that at all. But I will say that a small molecule has the most opportunity for immuno-oncology space using a really translatable type model. And then for large molecules, do I have a murine surrogate of the actual protein, or do I only have a humanized? So that's going to dictate what kind of animal model you can use. If you have a surrogate antibody, for example, you can use a competent mouse with a murine tumor, which again is more translatable than using an NSG mouse, which is completely immune compromised with a human tumor. That's just, and these would be more for ADCC like type antibodies. Other kinds of large molecule inhibitors, you can't really use like an NSG mouse with a human tumor. So another question is, what immune cells are required for my drug mechanism? Again, this is going to go back to what kind of animal model to use, which will dictate what kind of drug you need. So if you have to use a mouse model to get those immune compartments, you're going to want a murine surrogate or a small molecule. And if you don't have either one of those, then you need to find ways to piece together all of the data from different assays to tell the story that your humanized, large molecule or your cell therapy will actually do what it's supposed to do and have that interplay with the rest of the immune system. Is the mechanism of your drug translatable between human and murine immune cells? So again, if you're using competent mice, if what you're seeing with the Tregs of those mice, is that translatable to what's going to happen in humans? And the same thing goes with say using humanized mice. So if you use like hematopoietic stem cell reconstituted mice or PBMC reconstituted mice, sometimes those immune cells aren't actually working the way that they should, or maybe are a little bit malfunctioning in some ways. So it's important to make sure that the immune cells that you're working with are, recapitulate kind of the human situation as much as possible. And then what additional attributes does my drug have that contribute to the mechanism? So again, with like cell therapies, especially the engineered cell therapies, you might have a cytokine stimulation on that cell, or you might have a safety switch. Do I need separate assays to test these? And is in vitro or in vivo assay more appropriate to test these? And I will say most of the time for these proof of concept type studies, that in vitro is usually okay to test those attributes. You don't necessarily have to do in vivo for IND enabling, but it does depend on exactly what the attribute is. So when it comes to evaluating safety, you want to think about, are there any drugs with the same mechanism that have been that have published clinical safety data? Because that will guide you in actually designing your safety studies. And it will actually tell you whether or not you have to redo some of the stuff that's already been done. So you're going to want to compare, if you find it, you're going to want to compare your dose level, frequency, and the PK. Were the adverse events patient-specific or broad? Were they attributed to mechanism of action? And were they manageable? Like, back in the day when CAR Ts first started, CRS was a big problem. It's not that it's not a problem now, but we know how to manage it. So when people are getting infusions of CAR Ts and they're in the hospital, doctors are ready with antibodies and other treatments to treat CRS if it should pop up. And use what is known or what has already been learned to design your studies. Consider the proposed clinical protocol and attempt to emulate that when you're evaluating your non-clinical safety. So for example, dose scaling can be done with small molecules, to some extent large molecules, and definitely cannot be done with cell therapy. So the amount of cells you put into a mouse cannot translate to the number that you're going to put into a human. But that's okay. What you do want to make sure is that when you're running your IND studies, that you're doing things as close to what you'll be doing in a person as possible. So for example, if you're giving a cell therapy weekly for 3 doses, you want to run your tox study and your pharm studies with weekly dosing for 3 doses if possible. If you have to dose Proleukin in the clinic to keep your cells going, you're going to want to dose Proleukin to those animals as well in the safety and pharmacology studies. And then make sure, again, to focus on the drug attributes or engineering that might exacerbate or facilitate toxicities. And I'll get into that a little bit more in the following slides. So I'll bring up 2 case studies to compare 2 things that are very different. So a small molecule and a cell therapy in the IO space. So I'll talk about X4P001, which from X4 Pharmaceuticals, which is in the clinic right now. And it's a CXCR4 inhibitor, excuse me, that decreases recruitment of immunosuppressive cells, which express CXCR4 into the tumor microenvironment where CXCL12 is likely expressed. And then in doing that, it'll increase cytotoxic T cell infiltration. And then the cell therapy I'm going to talk about is a CAR-NK cell therapy from Katie Rezvani, which is in the clinic right now through MD Anderson. And the mechanism of action is killing of CD19 positive tumor cells by NK cells that have a CD19 CAR. Additional attributes to these cells is an IC9 safety switch, and then secreted IL-15 to help the NKs persist. Excuse me. So again, going back to our triangle, let's focus on the types of studies that we would need or things we'd have to look at for each individual drug. So it's a busy table, but I'll walk you through it. X4P001 on the left, and then the CD19 CAR-NK on the right. So what immune compartment of the host do we have to work with here? We need all the immune compartments of the host, especially myeloid-derived suppressor cells and Tregs, things that make a tumor cold and express CXCR4. And then for the NK cells, we don't really require any host immune compartment here. All of the immune cells are coming from the actual cell product. So for PK, as Kasha was saying earlier, it's driven by ADME, molecule size, formulation, et cetera, traditionally, we would draw blood and sometimes tissue to look at the PK preclinically. In a cell therapy, especially this one, the PK is driven by IL-15 and possibly cell expansion. The cells don't just necessarily clear the way a molecule might. So we would look at blood and tissue biodistribution and persistence over time. These cells might be hanging out upwards of months. So you would have to be evaluating this. And again, if they are expanding more, it's more exposure of drug to the system, and that could be an eventual safety issue, which I'll get onto later. So for safety, for small molecule, you can use a non-human primate, which is highly translational, and you would go through the traditional dose titration, acute tox, and MTD. Unfortunately, for a cell product, you don't get a lot of safety data because you can only use something that's severely immunocompromised, like an NSG mouse, not translational. You still are going to have to go through a dose titration, acute tox, and MTD. And then because of the additional attributes of these cells, like IL-15, you're going to want to also look at IL-15 secretion, like what kind of additional IL-15 is actually circulating in these mice, because that might have a upstream effect to people as far as activating and causing proliferation of the host immune cells if there's a lot of IL-15. You'll have to look at autonomous proliferation of the cells, meaning do they ever die? Do they ever go away because they have this IL-15 transgene? And also leukemic transformation, which some people don't think about, but NKs, when proliferating at a high rate and with constant exposure to IL-15, can turn leukemic. And then you might have to look at immunogenicity if in vitro, if possible. This depends on kind of the attributes of your cells. For pharmacology, for a small molecule or this specific small molecule, you'll and this is for efficacy and PD, you can use an immune competent mouse with a mirroring tumor, and that's great because then you can, like I was saying, you can really look at all the immune compartments on how they're interacting with this drug and prove your mechanism of action with a mirroring tumor. You can also do CXCR4 positive tumor cell growth inhibition and inhibition of colony forming units of immune cells that express CXCR4. For the NK, you'll be looking at an immune-compromised animal, NSG mouse with human CD19 positive tumor, and literally looking at tumor growth inhibition. Again, the tumor burden and the number of cells you're putting into this mouse is not translatable to the clinic. In vitro, you can look at serial killing or serial stimulation assay, which tests the persistence and the tumor killing capacity. And then for POC, there's not much other POC to do with this specific small molecule. Again, you can look at migration and chemotaxis assays just to look at the CXCR4 CXCL12 axis interplay. And this can be done in vitro or in vivo for this specific molecule because actually there's over 90% homology between mouse and human for this axis. That's not the same for other chemokine axes. But for the NK, because you have these additional features, you're going to want to look at long-term persistence, which does go hand in hand with both the PK, but also, again, the autonomous proliferation or potential for even tumorigenicity. And then you want to look at IC9-induced apoptosis using a dimerizing drug, and that could probably be done all in vitro, and in vivo is nice to have. So when you're engaging the FDA, you do want to, you can schedule an interact meeting, and this can be done well before you plan to file your IND. You're going to want to have your non-clinical study plan, and you can actually include data from pilot studies where you use either a biosimilar or a non-GMP drug to support the questions that you're going to ask. And the interact meeting is a chance for you to actually ask questions about the pharm tox studies as you design them for IND enabling studies, your manufacturing plan, release assays, your clinical plan. And the question format typically is you want to put out your proposal for what you want to do. And then you like to make the question a little bit vague because if you ask a detailed question, they can give you a detailed answer, but it doesn't tell you anything else about the rest of your proposal. So what we've done before is do a study proposal and say, does the FDA have any concerns with this approach? So then the FDA will write back typically any concerns that they have with the proposal that you wrote. Sometimes you don't get anything, but you're hoping by having a more broad question like that, that the FDA can actually look hard at your actual full proposal and then come up with any questions about any little piece of that. And then to support your study proposal and your question, you're going to have a summary of supporting data as well. And again, that can be generated with a biosimilar or non-GMP batch. Then there's also a pre-IND meeting. And a pre-IND meeting is not really an opportunity to ask questions so much. It's more, you are then proposing things. You're not asking questions about what you're doing. You're saying, okay, this is how we're going to do it. They may or may not give you feedback. But when you're doing your farm tox study proposal, you'll actually have study concept sheets, which are actual study designs. So talking about the number of animals you're going to use, the dosing regimen, exactly how many cells or how much drug you're going to be dosing, what the manufacturing plan is, release assays and clinical plan. And the idea here is, you're going to put this forth, propose it. Does the FDA agree? Yes or no? They might come back and say no, but then you don't know what they don't agree with. But typically by this point, you have a lot of confidence in what you've done. And, again, if it's a new class of drugs or something that's never been done before, you will have hopefully had an interact meeting ahead of time which would have given you more information about what they're thinking. So just some key takeaways for today. The FDA is primarily concerned with safety and efficacy. So when you're running your studies, keep that in mind. You don't have to be elaborate with your animal models necessarily if the data is going to be fuzzy or not exact, or you don't know what the metrics are for your readout, whether it's a go or no go. So it's better to be more poignant with what you're asking with the models that you're choosing. The IND enabling studies should interrogate all attributes of your drug. So if so you want to look at, again, mechanism, target, and any additional features that your drug might have. For tox, it's best to use as relevant or translatable assay or model as possible. But again, it's not always feasible, like in the case of cell therapies and NHP mice. So you have to lean on precedent. And in the case of cell therapies, especially CD19 cell therapies, there's tons of precedent. Right now, it's a very crowded space. But again, if you don't have that precedent, you have to do the best you can with bits and pieces of the assays to try to put the story together. And you need to allow several months for documentation and FDA feedback if you're doing interactive pre-IND meetings. So just keep that in mind. It's not a very fast process. And I think that's all I have. So I will take any questions that you have.
Thank you very much, Hilary. That was very informative. We have a couple of questions, but I wanted to ask one myself here first. When discussing, and you talked a little bit about the discussions with the FDA there at the end. I would understand that, like with a small molecule research and trying to get a small molecule inhibitor through with the FDA is a pretty cut and dry situation because we have, you know, decades of approvals and processes basically in place. But when it comes to like cell-based therapies where there is so much ambiguity, is that a much slower process with the FDA, or is it more streamlined now? I just wonder because there's so many different types of therapy and so many different MOAs, et cetera, et cetera.
Yeah, and I think it does depend on the target and what type of cell you're dealing with for your cell therapy. So again, like CD19 CAR T, for example, has been so well studied, and there's so much out there already that if you were to try to do another one, I think it would be fairly quick process. If you're doing a brand new therapy or one that hasn't been studied before, like say a gamma delta T cell towards you know, that was iPSC derived towards a solid tumor target or something like that. That would be a longer process, and you definitely, in my opinion, would want to have like an interact meeting ahead of time to make sure you're covering all your bases. So I think it does depend on target. It does depend on cell type. It depends on where your cells are coming from, like an autologous versus allogeneic versus stem cell derived. So yeah, it's a little bit all over the place. I'd say the more information that's already out there based on the cell and the target, the faster the process will be. If there's less information out there, you're going to have a little bit of a longer, harder time probably.
Okay, thank you very much. On the question on the Interact meeting there, we have a question from Sam again. What is your experience in getting an Interact meeting request accepted by the FDA?
So we've had them accepted, no problem. In my experience, I don't think we've ever been denied, and this was even during COVID. So I think it's a process. So I think if, again, you have a very novel drug and you have very specific detailed questions where that information is not available from any other source, they are very much willing to connect with you. But it is definitely all in how you write your meeting request. I will say that it's a kind of a, I think it's not for today, and I'm sorry, but please, if you need any help, please reach out to me on LinkedIn. I'm willing to kind of walk you through my experience with that.
Very generous of you, Hilary. I have a question here from, no, maybe we can take that later on in the group one. Ronald wants to make the point here that he would like to confirm that the price, wait, hold on there. Want to confirm that the price of out-of-pocket for non-clinical studies, i.e. preclinical studies, is about 600 million. This is because I understand a problem saying that the entire drug discovery process is 2.2 billion USD, but that includes the cost of opportunity. So the real cost is about 1.2 billion. Okay, thank you very much for that, Ronald. Sam just has a follow-up on your point there with the Interact, that from his experience, novelty is critical. If more standard drug designs, they are rarely approved. Okay. So now I would like to ask all of our speakers maybe to put your cameras on and join us if there are... Oh, sorry, Martine, you had a question for Hilary?
Yes, I did. Sorry, it's just... It's like slightly next to what you're saying, but when you are doing, for example, CAR Ts, I mean, especially cellular therapies, and you want to manufacture, let's say, the first batch for phase 1 or phase 2, I just want to confirm or like, I just want to know what you know about that, because I heard there is actually a problem in allowing different like new batches to actually go to phase 2 or phase 3, because there is I'm not sure, that's why I'm asking. There is apparently a quality control issue, since it's hard to judge without repeating all the studies with every single batch of, let's say, for example, CAR Ts, that they are actually the same, will have the same efficacy. And is there a way of doing that without repeating all the studies? Is there a standard for CAR Ts, or is it still, I would say, in development?
I don't know that there's a standard, but I think you just have to show your release assays. I can't imagine you would have to repeat every one of your studies. That would be a real holdup to any clinical progress. So I imagine that in your release assays, if you show that everything is within a certain range of variability, accepted range of variability between the batches, that should be pretty good. You might have to do a couple in vitro assays, but I can't imagine that you would actually be expected to repeat all of your non-clinical assays, because that would literally be like applying for another IND. Yeah. and that it's not feasible, and that it's too much of a holdup for companies. They wouldn't do it. So I think in my experience, we've had several batches, and we've actually moved from one manufacturing site to another. So we've done some very simple in vitro assays, and also done our release assays with both, and compared the results to make sure that they were within the variability, and it's been okay.
Okay, so no animal studies were necessary?
No.
Okay,
no.
Thanks a lot. Thank you.
Sure.
Thank you very much, Martine. So I'd like to ask our other speakers, Michael and Kasia, if you can turn your cameras back on and your audio, and we can maybe pitch some questions to the panel here. So we have a question here from Ronald, which I think you can all have an answer. Maybe we start with Hilary first, then Kasia, then Michael, go in reverse order of the speakers here. What is your opinion on 3D cell models and organs on chip as replacement for animal testing? So far, there is only one success case. That's Sanofi got drug approved for IND using only data from an organ on a chip developed by Hesperos. No animal data use. So the question is, what is your opinion on 3D cell models and organs on chip as replacement for animal testing? So Hilary, we can start with you.
Oh, okay. So again, I think it has to go with the novelty of the drug. I'd be shocked if this was a completely novel drug and that was accepted. I think it's okay. I think it's fine. I don't think there's any reason to use animals if it's not necessary, and if the drug has been tested, or the mechanism of action has been tested extensively elsewhere. And I think the FDA is in more of a position to save animals and to not do extensive animal testing if it's possible. So Yeah, I think it again goes back to novelty of the drug and the mechanism.
Okay, thank you. Kasia?
Yeah, so I think I will echo actually Hilary here, because I believe that there must have been some precedent actually before that. So I think it's all about the confidence. You have to be confident enough to not only have the approval of IND for first in human, but also you need to be confident enough to expect efficacy actually in the clinic, right? So there are also two separate things, right? So I think that the level of complexity is increasing with the complexity of the organisms that is being used for the testing. And as such, your confidence in the success of the clinical trial is also increasing. So, IND approval is one thing, but then also the success of the phase 1, phase 2, and phase 3 is a little bit different story. So depends, I guess, on the data we have available.
Okay, thank you very much. And Michael?
Yeah, I think that now as they're getting more complex, those non-animal models have a good place in preclinical development, to be honest with you. I think they can replace a lot of the expensive animal studies that are being done. Again, I think it goes back to the question that's being answered, whether or not they can be fully replacement for the animal models that they're predicting to replace. I think they have their place in preclinical development. And I think time is telling whether or not they actually are predictive or not of what will actually be seen in the clinic. But I think they are an alternative that can be used if you're asking the right question for the model that's to be used.
I think that's a fair comment from all of you. I think for me, at least anyway, the big drawback with using organ on chip and 3D models currently is the setup of the system, right? So it's typically 96 well plate format, and you put your drug on there, you leave it sit there for maybe 3 to 5 days. And in my opinion, does that really replicate what we see in the tumor microenvironment? And even just taking tumor interstitial fluid pressure in particular models and COP into account, whether you're going to get that level of exposure in a tumor compared with a 3D model or an organ on chip. So I know that there's a big move, particularly within the EU, to move away from animal models, but my feeling is that we're not there yet, basically, to be able to have that confidence going forward. Okay. Right. We had another question earlier on. I think that this was more relevant towards Michael's initial talk, but I think most of you have worked with developing humanized models, so maybe you can all weigh in, but we'll start the answering this time with Michael. So the question is related to PBMC donor variation. And in your talk, Michael, you presented that you'd use the same PBMCs for the subcutaneous and orthotopic models, which was very elegant. And the question here basically was, where is this again? The immune cell recruitment to the tumor microenvironment was different between the subcutaneous and the orthotopic. And I just wondered if you could maybe elaborate on why that is, and if you can maybe expect the same type of answers from both of them.
Yeah, so I think that's kind of our point too, is why we think that implantation site matters, is that the recruitment is going to be different just based off where the tumor is and where the vasculature of the tumor, the ability for cells to get to the tumor and actually to have an impact on the tumor, right? So subcutaneously, that's a little bit easier because they are a little bit more vascular. And they are a little bit easier to get to than if you say you put it orthotopically. And so, yeah, I think that's kind of why we're seeing differences and why we think it's important as well, too, because when you model a subcutaneous tumor, you're not really necessarily showing what it would look like if you did it inside an organ where you'd actually see the tumor in a clinical setting. And so they are more intricate and more advanced models, and that does come with a cost, but it also comes with, we think, greater clarity and greater insight to what you'd actually see when you move into the clinical setting. Because as you've all seen and everyone probably knows, it's very easy to we've seen great therapeutics, immuno-oncology therapeutics in vivo in mouse models, but like I said, they haven't been very good in humans. And why is that? And I just don't think that we're looking at them in the right setting based off of microenvironmental interactions and the dynamic stromal interactions that you see for IO agents that are required. So I think there are differences in there, and that's why it's kind of important to think about that when you're designing your preclinical experiments.
Yeah. Kasia?
Right, I'm not sure if there's anything more to add. It's just that from my side, I think, because I was actually using not PBM system, but actually CD34, not very extensively though. And I would say that it would be good to also run sort of like a time course study, which actually, to be honest, I'm not aware that there is a good publication actually on it. Probably also because just of the costs of this kind of studies, to really understand whether the infiltration in the tumor is actually increasing, whether it's changing at all. Another aspect is that still these models, I know that they're being improved over time. There are now different models available with some sort of like cytokines booster.
Okay.
And etc. Another thing is that in severely immunodeficient animals, the thymus is nearly absent. So this is actually another thing that is kind of like obstacle for the T cell maturation and education as well. So this is another problem here. From my experience, what else I can add? Yeah, so those models, in my opinion, are still imperfect, but as Michael also said, depends what kind of question you're asking at the time. So I think one question at a time is good always.
And Hilary? Can you hear us, Hilary? We can't hear you.
Yeah, sorry about that. My AirPods just died right in the middle of this. Can you just repeat the question one more time? Because I think I heard two different things.
Yes, let me just find the question again. Yes, it was about the PBMCs that were used in Michael's presentation, subcutaneous and orthotopic. And basically, we wanted to know about the immune cell recruitment into the tumor microenvironment was quite different between the subcutaneous and the orthotopic levels. And could you comment on that and its relevance towards testing of drugs?
Yeah, sure. So I think I agree with what Michael was saying earlier, because I have some experience with this from J&J. We did a lot of this work, but most of our work was around the sub-Q tumor model with PBMCs and not orthotopic as much. But the sub-Q, most human tumor, I wouldn't say most, so at least a good handful of them have very poor vasculature. So I think you don't necessarily get the recruitment of the immune cells that you necessarily want when you want it. So, doing a time course to look at the sub-Q tumor as far as when you have, if you need T cells, cytotoxic T cells there, when you have the T cells infiltrating, whatever, that's probably your best bet. It is donor dependent, and but ideally you actually screen your donors for their phenotype, their immune phenotypes before you actually put them in, or you do at least an engraftment study for PBMCs. I don't have too much experience with the orthotopic, but I think, especially with those tumors, you probably have a lot more vasculature and better infiltration of immune cells.
Yeah, I'm just wondering as well, between all three of you, one of the things that I think is maybe overrepresented when it comes to the humanized models is people looking for TILs, right? In particular, and trying to define what those TILs are in humanized models. However, I think one of the biggest caveats of the humanized system is the lack of representation of the monocytic population. And in particular, I just wondered, have you guys looked either with PBMC or CD34 humanized models, what sort of representation you get of MDSCs or the myeloid populations intratumorally in either orthotopic or subcutaneous models? Have you been able to model that? What is the persistence, particularly in PBMCs, of myeloid cells, and are they functional at the tumor site as a resistance mechanism?
Yeah, so I could take that one to start. You're right that the PBMC engraftment ones are really mostly used for T cell therapies. Most of the repopulation that you get there is strictly driven by T cells, and that's why we didn't look at any of the monocytic populations, to be honest with you, because they're not really there, and if they are there, they're not in any population that would be beneficial. There are alternatives, though. I think someone mentioned this earlier in their talk as well, too. I forget if it was Hilary. But there are other humanization models that are coming that are using different cytokine stimulation that can actually look at the monocytic population. So there are, but those are more CD34 engrafted models that have different levels of cytokine activation. So they actually have a monocytic population. And we've actually done some studies with those for clients as well too. And they do have monocyte populations, so you can actually look at those different types of therapies as well too. Because as you mentioned, not all therapies are T-cell based anymore when it comes to IO therapies, right? There is a monocytic population that is important there, and that are designing therapies around those. So having the model to check for those is important. And yeah, the PBMC ones aren't really good for that, but there are other alternative models for humanization that can be done on that.
Just to interject before we go on to Kasia and Hilary, Michael, you mentioned there are models now with cytokines. Are you talking about the NSGS, or are you alluding to different models here?
No, those are the ones I'm talking about. So the vendors are making models that are specific for, like, so JAX and Taconic and things like that, the NSG, SGM3, or NOG, EXLs, depending on the background that you like to use. And they have done time course studies to show that they do get a good population there, and that it is persistent over time. So yeah, those are the types of models that I'm talking about. Yeah.
Okay, thank you. Kasia or Hilary?
Yeah, so yeah, we didn't really have actually best experience when it comes to CD34 implanted animals and response to PD-1, so we have not really seen much of a response. So it seems that the myeloid compartment is not exactly functional, and we don't really have a very good antigen presentation there. So definitely it's problematic. My colleagues from another group will be also trying the model with it's genetically engineered to express GM-CSF, and let's see how that one goes. There are also some vendors that are providing animals, or at least sort of like cytokine cocktails that you could potentially use depending on the cell population of interest. But of course, then the question is, what is the best time for injection of such cocktail, or whether you have to actually do repeat injections, etc. Right? So it's not also as straightforward as it would be. So yeah, it's I would say it's still a work in progress, and in my opinion, there's still quite a bit that has to be done, and in order to have these models really useful, and kind of like from my experience and also from other groups that we have here, it seems that actually you still have to validate these models internally and really understand them, really understand the timing, the best timing for injections, for the treatments, for implantations, etc. So it's still, yeah, no, it's still a long way, I would say.
Yeah. Hilary?
Agree with all the above. I think kind of there comes a point where you have to say to yourself, this is a mouse and it's not going to be human. And it's eventually turning into a Franken-mouse. And then what does your data mean afterwards? So I think, especially, and I look at it, maybe this is with a regulatory perspective, certain things you might not have to go there. It might be a lot of trouble, a lot of expense, a lot of time for something that you don't need, that might give you answers that you're not so sure about because of whether or not the immune cells are functional, whether or not you have enough of them. Is the amount of immune cells that are there in the tumor anywhere near or the functionality or the number that you would see in a human? So, again, for POC, like the T-cell work, bispecifics, things like that, they're great. Humanized models are great for that. But yeah, certain populations, they're not wonderful. We've gotten okay with B cells. We've gotten okay from PBMCs. Myeloid compartment is trash. It's going to be a lot of time and a lot of work to get it there.
Yeah. I saw there was a recent paper published where they used PBMCs derived from cord blood to try to improve these populations. I also have seen another paper where they have attempted to use iPS reprogrammed CD34s in this situation. Have you guys used any of these approaches? No,
I haven't.
No.
And that would lead on then to my question as well, as regards the use of preclinical within your organizations. Obviously, Michael, you're a vendor. You're working specifically with models on a day in, day out basis. But When it comes to your experience from partners, and then also from Hilary and Kash's point of view, working within the pharma industry, how much does pharma put into its preclinical development platforms? Are you actively looking at building up these areas, looking at the 3D models and organs on chips as surrogates, or is there a wind down on the preclinical development, particularly as it's relevant to immunotherapeutics, given that the authorities are now saying, you know what I mean, that we can get something into the clinic without having it go through an animal at all? What's your feeling, and what is the status of your companies and their experience within this area? We'll go with Michael first.
Yeah, I was gonna say, happy to jump in and take that one. Yeah, I think that you mentioned earlier when we were talking about that 3D organ on chip. I think that there is a push to move those things there, but I don't think it's there yet. I don't think we're away from animal models at the moment. So I still think that animal models do play a specific role in preclinical drug development. And from the pharma partners and the biopharma partners that we're working with, we do see some that are actively looking to build new models and more extensive models for the questions that they have answered. So, you know, there is an active engagement with clients that we're working with that have the need to build specific animal models still, and wanting to do that, and more complex models as well too, right? They want models that are more representative of a human population that they're going to be using when they treat in the clinical trial setting. And so that's looking for models that are heavily pre-treated with agents that you'll see in the clinic. So they don't want any naive PDX models. They want models that have been treated with, you know, standard of care a couple different times, so they can actually, you know, model what they're going to see for their first line therapy when they go into their phase 1 clinical trials, right? So there's a lot of that. So I would say, you know, even with all the news in the biopharma and the preclinical development, a lot of preclinical development companies getting cut, that's usually the first thing to go so people can focus on their clinical candidates. There is still the need for animal models and models that are representative of the population that they'll see when they go into the clinic. So, you know, that's kind of my thoughts on it and what I've seen so far and recently.
Great stuff. Thank you very much. Hilary?
I think that there is definitely still a need for animal models. I struggle sometimes, like, for example, with the cell therapies when we have to run tox studies, for example. There's nothing that says any of that tox is going to translate because the dose isn't translatable. And there's nothing for those cells to interact with in a mouse. It's all, you know, it's just a tumor. And when you're doing a non-tumor bearing tox, I don't understand why it's still necessary. So there are things like that. And I think there's enough push, at least from the cell therapy world, to try to change that. Now for other drugs, where you have more translatable models, both for efficacy and toxicology, like competent mice, or you can use an NHP, I think that's definitely necessary, and I don't think we're going to get away from that anytime soon. It's really tough. I think for efficacy and POC, still need the animal models. For tox, I think it can be called into question depending on what your drug is.
Thanks, Hilary. And Kasha?
Yeah, so I think that there is definitely still and always appetite for innovation, and at the same time there is the notion to limit animal experimentation, but I agree that we are not really able at the moment to really get out from there. So first of all, we have quite a bit of problems translating in vitro to in vivo. That's one thing, and then from in vivo to the clinic, it's actually another level of complexity. So translating directly from in vitro to the clinic would be definitely difficult. We have actually the best experience with running so-called mouse clinical trials. So we've been using extensively PDX models for this, and we actually have quite a long history of running the MCTs, and there's actually quite old publication also coming from Novartis that I can invite everyone. Some of these drugs that were actually tested back in the days when the publication was coming out, now they're actually in the clinic. They're actually marketed drugs, so you can actually compare some of the results. So this is actually quite intriguing that there is some resemblance actually of the results. But as I said, from in vitro to in vivo and then in vivo to the clinic, it's a little bit difficult. And also, as Hilary said, the tox, I mean, you cannot really replace that, right? So here, animal testing has to be still involved. This is something that we cannot really avoid at the moment, at least.
No, completely agree.
We can minimize, but not avoid.
No, exactly. So, and then maybe a follow-up question here. This has come from Nicholas Dietes. When it comes to in vivo testing, what is the relationship of IC50 you expect to see between in vitro and in vivo efficacy?
Oh, this is a bit more complex. We need like another hour maybe to discuss this. So overall, kind of like ballpark, I think that the values in the low nanomolar, tens of nanomolars in high-bar models, maybe low hundreds of nanomolars are kind of like good start, and we can potentially actually kind of like formulate it in a way that it will be those first to mice and then to patients. But the truth is that it's not as easy, because first of all, you need to also understand what is your dependency between the PK and PD, what level of target engagement you need to reach, what kind of level of PD modulation you need to reach. So for certain targets, you are also able to set your downstream PD marker and define whether it's actually IC50 that is needed. Maybe it's IC20, maybe it's IC90. It's hard to say. Another thing, when you go to animal models and then also to the human, you also have plasma protein binding. You will have exposures. So it's I'm afraid not as straightforward. but I would say start with sort of nanomolar levels.
Okay, thank you very much. Hilary or Michael?
I have no opinion. I'm going to defer to Kasia.
I think that was a pretty impressive answer. Yeah.
Great stuff. We don't have any more questions here. Anybody else on the call have any questions? Please just raise your hand or forever hold your peace. Okay. We don't seem to have any more questions coming in. So then I would like to wrap up the call. I'd like to, first of all, thank our speakers very much for joining us, particularly Michael and Hilary getting up so early in the States and joining us very early hours of the morning, particularly for Michael, who's on the west coast of the US, and Kasia as well as our sole European speaker holding up the fort for us here. Thank you all very much, not only for your presentations, but also your contribution towards a very nice discussion. I'd also like to thank Barbara and Rosalinda from Immunomodel for setting up the webinar, and to everybody who joined us here today and asked questions to make this an interesting webinar. As Hilary pointed out, if you have questions, etc., just reach out to her on LinkedIn, and I presume the same can be said from Kasia and from Michael. If you have any direct questions about the webinar or anything like that, please contact me, and I will get back to you. And I believe, Rosalinda, this webinar has been recorded and will be available on the Immunomodel website for those who have registered.
And also on the social media and the YouTube channel, so it's available for whoever wants to listen to this webinar subjects. So we are very happy to share our activities as an Immunomodel activity and cost action. So thank you very much.
Okay. So with that, I again would like to say a final thank you to our speakers, to everybody who joined, and yeah, thank you and enjoy the rest of your afternoon.
Thanks for being
Thank you all.
Bye-bye.
Bye.
I'm